At some point during the 14th or 15th hour of a long working day, something shifts. It is not simply that the body aches, or that concentration becomes harder to sustain, or that the desire for sleep grows from a mild preference into something that feels closer to physical pressure. It is that the body itself seems to have changed its fundamental properties. The arms, which at 8 in the morning required no particular effort to lift, now feel as though they are made of something heavier than they were before. The legs carry their own weight with a reluctance that approaches protest. Standing up from a chair, an action performed dozens of times a day without any conscious expenditure of intention, becomes a negotiation between mind and body, a process that requires the kind of deliberate recruitment of effort normally reserved for tasks of actual physical difficulty. The geometry of the body seems to have subtly altered. Gravity, the constant and indifferent force that presses everything in the universe toward everything else, appears to have increased its claim. It has not, of course. The gravitational constant, the number that governs the attraction between masses, expressed in Newton's law of universal gravitation as the product of two masses divided by the square of the distance between them, multiplied by a constant of approximately 6.674 times 10 to the negative 11th power, in units of cubic meters per kilogram, per s-econ squared, does not fluctuate with the hours of the working day. The Earth has not grown more massive. The body has not increased in weight. The physics of the situation, in the strict mechanical sense, is entirely unchanged between morning and evening. The mass of the body, pressing against the floor, is identical. The gravitational acceleration at the Earth's surface, approximately 9.8 meters per s-econ squared at sea level, is for all practical purposes exactly what it was when the alarm went off. And yet the experience of heaviness is real. It is reported consistently across populations, across cultures, across the full range of conditions that produce fatigue, physical exertion, sleep deprivation, cognitive labor, immune activation, emotional stress, chronic illness. It is not imagined in the dismissive sense, not fabricated or confabulated, not a psychological artifact of low mood or self-indulgent complaint. It is a genuine perceptual experience with identifiable mechanisms, produced by real and measurable changes in the biological systems, responsible for generating the felt sense of the body's weight, and the effort required to move it through space. The disconnect between the unchanging physics and the dramatically altered experience is itself the most interesting thing about fatigue, because it reveals, with unusual clarity, the degree to which the body that a person inhabits from day to day is not a physical object directly sensed, but a model, continuously generated by a nervous system, that is, like any model, vulnerable to the conditions under which it operates. To understand what actually happens when the body feels heavier under fatigue, it is necessary to begin not with the muscles, though the muscles are crucially involved, but with the question of how the brain knows what the body weighs in the first place. The answer is less obvious than it might appear, and it illuminates something fundamental about the relationship between physical reality and biological experience that extends well beyond the specific phenomenon of tiredness. The brain does not have a mass sensor. There is no organ in the body, no dedicated neural structure that simply reads the body's weight and transmits that value to consciousness the way a bathroom scale transmits a number to a display. Instead, the felt weight and mass of the body is a construction, an inference the brain makes continuously from a rich and complex stream of signals originating in several distinct sensory systems, integrated and interpreted through a hierarchical neural architecture that is itself subject to the same constraints of energy, resource allocation, and accumulated biochemical change that affect every other biological process. The primary contributors to this construction are the muscle spindles and Golgi tendon organs distributed throughout the body's musculature, specialized mechanoreceptors embedded in the tissue of skeletal muscles and tendons, respectively, each responsible for reporting different aspects of the mechanical state of the muscle to which they belong. Muscle spindles are stretch receptors. They detect changes in the length of a muscle fiber and the rate at which that length is changing, providing the central nervous system with continuous real-time information about the position and movement of every limb. Golgi tendon organs, positioned at the junction between muscle fibers and the tendon through which the muscle exerts its force on bone, detect the tension being generated by the muscle during contraction. The actual force the muscle is producing at any given moment. Together, these two receptor populations provide the brain with the data from which it constructs the proprioceptive sense, the awareness, largely implicit and below conscious attention, of where each part of the body is in space, what posture it is maintaining, and how much force is being exerted. The sense of heaviness, the felt weight of a limb, is generated substantially through the integration of this proprioceptive data with a second stream of information that is less often discussed, but equally important: the signal arising from the brain's own motor commands. When the brain instructs a muscle to contract, it does not simply transmit the command to the muscle and wait for sensory feedback. It simultaneously generates an internal copy of that command, a signal called the efference copy, or corollary discharge, which circulates within the central nervous system and is used by higher brain regions to predict what the sensory consequences of the motor command should be. The predicted sensory feedback is compared with the actual sensory feedback arriving from the proprioceptors. The difference between them, the prediction error, is used to update the brain's model of the body's current state and to calibrate subsequent motor commands. This system has a consequence for the subjective experience of effort that is both elegant and under conditions of fatigue, profoundly consequential. The perceived effort of lifting a limb or sustaining a posture is not simply a readout of the mechanical force the muscles are generating. It is substantially a readout of the magnitude of the motor command the brain had to issue to produce that force, of how loudly, so to speak, the brain had to shout at the muscle to get it to do what was required. Under normal rested conditions, a relatively modest command produces a normal muscular response, and the effort feels proportionate to the task. Under fatigue, when the muscle's capacity to respond to neural commands has been degraded by the accumulated biochemical consequences of prior contraction, the same task requires a larger motor command, and the effort feels correspondingly heavier. Even though the physical weight being moved has not changed at all, the biochemical degradation that drives this effect is not a single process, but a cascade of interrelated changes unfolding at the cellular level across every working muscle fiber. The contraction of a skeletal muscle fiber depends on a series of events that begin with an electrical signal from a motor neuron crossing the neuromuscular junction, the specialized synapse between nerve terminal and muscle fiber, releasing the neurotransmitter acetylcholine, which initiates an action potential that propagates along the muscle fiber membrane and triggers the release of calcium ions from an internal store called the sarcoplasmic reticulum. The calcium ions diffuse to the contractile proteins, actin and myosin, enabling them to form cross, bridges and generate force through a cycle of attachment, power stroke, and detachment that is powered entirely by the hydrolysis of adenosine triphosphate, the cell's fundamental energy currency. Each power stroke consumes one molecule of ATP. The force generated by the whole muscle is the summed output of billions of these microscopic mechanical events occurring simultaneously across thousands of fibers. The problem is that ATP is not unlimited, and the metabolic pathways through which it is regenerated during sustained muscular activity produce byproducts that progressively interfere with the very processes they are meant to sustain. Phosphocreatine, the muscle's immediate reserve of high-energy phosphate groups used to rapidly regenerate ATP from ADP, is depleted within the first 10 to 30 seconds of maximal exertion. Beyond that point, ATP regeneration shifts to glycolysis, the anaerobic breakdown of glucose, which produces ATP relatively quickly, but also generates inorganic phosphate and hydrogen ions as byproducts. The accumulation of inorganic phosphate directly interferes with the calcium releasing mechanism of the sarcoplasmic reticulum and with the force-generating capacity of the myosin cross-bridge cycle. The phosphate enters the cross-bridge and reduces the power stroke's mechanical output. Meanwhile, the declining intracellular calcium concentration means that not all contractile proteins receive the calcium signal necessary to engage, reducing the number of active cross-bridges contributing to force generation. The muscle, in a very precise biochemical sense, begins losing its ability to respond at full strength to the electrical commands it is receiving. This is peripheral fatigue, the failure occurring at the level of the muscle fiber itself. It is measurable, quantifiable, and mechanically direct in its relationship to the felt experience of heaviness. But it is only half the story, and arguably the smaller half. For alongside the peripheral changes in muscular biochemistry, a separate, and in some respects more fundamental process is unfolding within the central nervous system itself, a process that alters not the muscle's capacity to respond to commands, but the brain's willingness and ability to issue them. Central fatigue, the reduction in neural drive to muscle, originating from within the brain, rather than from changes at the neuromuscular junction or the muscle fiber, has been recognized as a genuine physiological phenomenon since the early 20th century. But the mechanisms underlying it have only been systematically characterized in recent decades. One of the most important contributions to this understanding came from a 2022 study conducted by researchers at the Paris Brain Institute, using magnetic resonance spectroscopy to measure the concentration of neurotransmitters in the lateral prefrontal cortex of participants who had been engaged in cognitively demanding tasks for several hours. The researchers found that the concentration of glutamate, the primary excitatory neurotransmitter of the central nervous system, and a molecule produced in abundance as a byproduct of sustained neural activity, accumulated progressively in the prefrontal cortex as cognitive labor continued, and that this accumulation was correlated with measurable changes in task behavior. Specifically, a shift toward less effortful strategies and a reduction in the willingness to invest further cognitive work in demanding decisions. The buildup of glutamate appeared to signal to surrounding neural circuits that the prefrontal region was operating at the edge of its sustainable workload, generating, in effect, a biochemical fatigue signal at the level of the brain itself. The significance of this finding extends beyond the cognitive domain. The prefrontal cortex is not merely involved in conscious deliberation and decision-making. It provides crucial descending input to the motor system, input that contributes to voluntary effort, to the sustained drive to continue a physical task in the face of discomfort and resistance, and to the brain's ongoing calibration of how much neural resource it is willing to commit to the motor commands driving muscular contraction. When the prefrontal cortex is operating under the constraint of glutamate accumulation, the motor commands it endorses and sustains become more conservative, not because the muscles cannot respond more vigorously, but because the cost-benefit calculation performed by fatigued prefrontal circuitry returns an answer that favors disengagement. From the brain's perspective, as researchers at the University of Brighton proposed in a 2025 paper, fatigue may function as a protective mechanism, a neurological governor that prevents the sustained overexertion of biological systems by generating an increasingly aversive experience of effort that motivates the organism to stop before genuine tissue damage occurs. The heaviness is, in part, a warning that the brain is generating deliberately through changes in how it processes and amplifies the signals arriving from a struggling musculature. This protective function becomes clearer when examined in parallel with what happens during sleep deprivation, the most common condition under which ordinary people experience the maximal version of the body, feels heavy phenomenon in daily life. The accumulation of a molecule called adenosine in the brain during wakefulness is central to understanding this dimension of fatigue. Adenosine is a byproduct of cellular metabolism in the brain. Specifically, it is produced when neurons consume ATP during the work of thinking, processing, and maintaining electrical activity. As the hours of wakefulness accumulate, adenosine builds up in the extracellular space of the brain, binding to adenosine receptors that are distributed across regions involved in maintaining alertness, sustaining attention, and motivating voluntary movement. The adenosine binding progressively suppresses the wake, promoting neural circuits, particularly those in the basal forebrain and the locus coeruleus, and inhibits the noradrenergic and cholinergic systems, whose activity underlies sustained attentional engagement and motor readiness. The experience of this chemical accumulation is directly familiar. It is the feeling of heaviness behind the eyes, the increasing friction in every thought, the quality of consciousness that begins to feel like moving through resistance rather than through air. What sleep accomplishes, among its many restorative functions, is the systematic clearance of adenosine from the brain. During sleep, the glymphatic system, a network of channels surrounding cerebral blood vessels that was only fully described in 2013 in work by Macon Niedergard and colleagues at the University of Rochester, becomes dramatically more active, expanding the space between cells in the brain and allowing cerebrospinal fluid to flow through the tissue and wash metabolic waste products, including adenosine and other byproducts of neural metabolism out of the brain and into the circulatory system for disposal. The morning alertness that follows a full night of sleep is, in a literal biochemical sense, the subjective experience of a brain whose adenosine load has been cleared, a system that has been reset to the beginning of its metabolic tolerance for wakefulness. The heaviness of late-night exhaustion is, correspondingly, the experience of a brain operating under the maximum sustainable adenosine concentration, its wake-promoting circuits suppressed to the threshold of involuntary sleep onset. But the heaviness of fatigue is not only generated by changes in the brain's neurochemical state, it is also produced by changes in the body's immune system. And this dimension of the phenomenon connects fatigue to one of the most ancient and conserved biological programs in vertebrate evolution. The experience of being sick, the aching heaviness, the reluctance to move, the withdrawal from normal activity, the sensation that the body has become an obstacle rather than a vehicle, is not a mere side effect of infection. It is a deliberately orchestrated behavioral state produced by the brain in response to immune activation, mediated by a class of signaling molecules called cytokines. When the immune system detects a pathogen, or, more broadly, any signal of tissue damage or physiological challenge, it releases pro-inflammatory cytokines, including interleukin-1 beta, interleukin-6, and tumor necrosis factor alpha. These molecules do not merely coordinate the immune response locally. They also cross the blood-brain barrier and act on the brain directly, or signal to the brain through the vagus nerve, inducing what has been called sickness behavior, a coordinated suite of changes in motivation, movement, and perception that includes fatigue, hyperalgesia, increased sensitivity to pain, social withdrawal, and the felt sensation of profound physical heaviness. The adaptive logic of this orchestrated heaviness is straightforward. An animal that is fighting infection conserves metabolic resources by reducing voluntary physical activity, diverts energy from muscular exertion to immune function, and reduces the risk of further injury or predation by withdrawing to a sheltered location. The heaviness is the behavior generated by the brain as a motivational intervention, a perceptual state engineered to make rest feel necessary and movement feel costly. What research has revealed in the past decade is that this same cytokine-mediated mechanism is active not only during frank infection, but during the routine metabolic stress of intense physical exertion, sleep deprivation, chronic psychological stress, and extended cognitive labor. Interleukin-6, in particular, is released by working skeletal muscle during prolonged exercise, a discovery that earned muscle the surprising designation of an endocrine organ capable of producing systemic signaling molecules that communicate the state of peripheral tissue to the brain. The IL-6 released by exercising muscle reaches the brain and contributes to the central fatigue signal, the perception of heaviness and the declining motivation to continue exertion that many people experience as the subjective limit of physical performance. The brain, receiving this immune signal from the body, interprets it through the same circuits that process sickness behavior and responds with a neurological state designed to promote rest. The body aches not only because the muscles have been chemically disrupted, but because the brain has been chemically instructed to make the body feel as if it has. The interplay between these peripheral and central mechanisms, the failing ATP regeneration and calcium dynamics in the muscle fibers, the accumulating glutamate in the prefrontal cortex, the rising adenosine load, suppressing arousal circuits, the cytokine signals translating peripheral exertion into central fatigue, produces the composite experience of heaviness through a final pathway that returns us to the proprioceptive system, and the question of how the felt weight of the body is generated in real time. Under fatigue, each of these mechanisms degrades the efficiency of the system in a direction that increases the discrepancy between the motor command the brain must issue and the mechanical response it receives from the muscle. The muscle spindles and Golgi tendon organs, themselves affected by the altered calcium dynamics and metabolic state of fatigued fibers, begin to report a different relationship between the neural signal and the mechanical output. The efference copy mechanism, comparing issued commands to their sensory consequences, detects that the commands are increasingly underperforming, that more signal is required to produce the same movement. The brain, integrating this information across the entire musculature simultaneously, arrives at a global upward revision of the body's apparent weight. The arms and legs feel heavier because the neural evidence, the ratio of command to response, suggests that they are offering more resistance than they were before. This is not an error of perception, in the sense of being a false signal. The resistance is genuinely greater at the interface of nerve and muscle than it was at the start of the day. What has changed is not the gravitational mass of the body, but the biological efficiency of the system that moves the body against gravity, the biochemical and neural machinery that translates intention into force. The physics of gravity is constant. The physiology that contends with it is not. And the brain, which must maintain a continuously updated model of how much effort particular actions require, adjusts its sense of the body's weight in response to the efficiency of the machinery rather than the mass of the object. The heaviness is accurate information about a genuine change in the body's mechanical situation, expressed in the language of perceptual experience rather than the language of engineering. This understanding has consequences that extend beyond the phenomenology of a long working day. It illuminates why fatigue is so poorly served by the cultural frameworks most commonly applied to it. In the dominant productivity-oriented framework of contemporary professional life, fatigue is routinely treated as a motivational deficit, a failure of discipline or commitment that can and should be overcome by the application of willpower. This framework is not only scientifically inaccurate, but potentially harmful, because it misidentifies the nature of the experience it is trying to address. Fatigue is not primarily a psychological attitude toward effort. It is a biological state produced by real changes in neural chemistry, metabolic biochemistry, immune signaling, and proprioceptive feedback. Changes that are not reversed by motivation, not circumvented by determination, and not suspended by the conviction that the work must continue. The heaviness is not a story the body is telling about how it feels about the situation. It is a report about the actual condition of the tissue and the neural architecture that animate it. This distinction matters in contexts far beyond individual productivity. In the practice of clinical medicine, fatigue is one of the most common presenting complaints and one of the most consistently under-investigated, frequently dismissed as a symptom too vague and subjective to warrant the diagnostic resources that more legible symptoms attract. The science of central and peripheral fatigue suggests that this dismissal is not medically justified, that the felt sense of heaviness and exhaustion is a genuinely informative signal about the state of multiple biological systems simultaneously, including the immune system, the musculoskeletal system, the glymphatic clearance function, and the metabolic state of the prefrontal cortex, conditions in which fatigue is the primary complaint. Myalgic encephalomyelitis, post-viral fatigue syndromes, including the post-COVID condition that has affected millions globally, fibromyalgia, and a range of autoimmune disorders, are conditions in which the mechanisms described here are dysfunctional in ways that remain incompletely understood, but that are increasingly accessible to investigation through the frameworks that the neuroscience and physiology of fatigue provide. The heaviness these patients report is not imagined. It is the product of real biological dysfunction, and understanding the mechanisms of normal fatigue is the necessary foundation for understanding why these mechanisms can become pathological. In the domain of athletic performance, the science of fatigue has driven a significant reconceptualization of what the limits of human physical capacity actually represent. The traditional model of athletic fatigue, developed partly from the work of physiologist Archibald Hill in the early 20th century, treated exhaustion as a purely peripheral phenomenon, a mechanical failure of the muscle when its biochemical reserves were depleted, or its contractile proteins were insufficiently supplied with oxygen. The modern model, associated substantially with the work of exercise physiologist Tim Noakes at the University of Cape Town, and his development of what he called the central governor model, repositions the brain as the primary site at which the limit of performance is determined. In this model, the brain continuously monitors the physiological state of the body during exercise, attending to signals from the muscles, the heart, the metabolic system, the thermal regulation system, and imposes a central fatigue signal that reduces motor drive and generates the subjective experience of exhaustion before peripheral mechanical failure actually occurs. The body, in this view, stops before it breaks. The heaviness that makes a marathon runner slow down in the final miles, or makes a climber feel unable to commit to the next move on a difficult route, is not simply the mechanical consequence of depleted energy stores. It is the nervous system's conservative estimate of the margin between current performance and physiological failure, translated into a perceptual experience compelling enough to motivate behavioral change. The implication is that the felt limit and the actual limit are not identical, and that the gap between them varies with training, with psychological context, with the meaning attributed to the effort, and with the brain's accumulated experience of how safely the boundary between exhaustion and damage can be approached. Highly trained athletes are not stronger in the simple sense of having more powerful muscles than untrained individuals. Though this is partly true, they are also more accurately calibrated. Their central nervous systems have learned, through thousands of hours of approaching the boundary between effort and exhaustion, to distinguish more precisely between the protective signal of fatigue and the genuine signal of imminent harm, and to sustain effort closer to the actual mechanical limit without crossing it. Their bodies feel heavy at the end of a race. But the point at which that heaviness becomes prohibitive is further along the performance curve than it would be for an untrained person, because the brain has refined its model of how much the experienced heaviness represents actual danger. There is something instructive in this picture for how the experience of fatigue is understood more generally, not as evidence of personal inadequacy or insufficient motivation, but as one of the most sophisticated information processing operations the nervous system performs. The brain is simultaneously monitoring the biochemical state of every major muscle group, tracking the ratio of motor commands to mechanical responses, integrating immune signals from peripheral tissue, managing the accumulated adenosine load of hours of wakefulness, weighing the glutamate accumulation in its own prefrontal circuitry, and translating all of this into a perceptual experience. The felt heaviness, that is proportionate, motivationally compelling, and designed to prompt exactly the behavioral response that will best serve the organism's long-term integrity. The experience is not a failure of any system to do what it should. It is the successful output of a system doing precisely what it was built to do, under conditions that push it toward its designed limits. What changes between morning and evening, between the body that rises without thought and the body that settles back into a chair with the quality of something yielding to a force it can no longer oppose, is not the physics of the world pressing on the body. The gravitational constant remains what it has been for 4.5 billion years. What changes is the biology that navigates that physics, the calcium dynamics in billions of muscle fibers, the glutamate concentration in the lateral prefrontal cortex, the adenosine burden accumulating in the basal forebrain, the cytokine signals traveling through the bloodstream from tissue that has spent its reserves, the efferent's copy system, revising upward its estimate of what each movement costs. Gravity does not become stronger. The body becomes, in precise and measurable and temporary ways, less capable of holding its own against it. And the brain, faithful to its function as a model, builder operating always in the service of survival, translates this capacity loss into the language that will most reliably motivate the right response. The felt, embodied, unmistakable experience of weight. The heaviness is the biology speaking. It is worth, at the end of a long day, learning to hear what it is saying.